So thanks very much. And those of you that are standing, exercise is good for you, so maybe you can move around a little bit. Brad will tell us more about that as we go forward tonight. It's my pleasure to serve as the chair of the Department of Kinesiology and to help organize this event every year. Clearly, the Pease Family Scholar Lecture is one of the highlights of our academic year in the department. This is the 16th lecture in the series. We're here tonight because of the generosity of Dean and Sally Pease and their lasting commitment to the Department of Kinesiology and Iowa State University. Dean Pease was a longtime faculty member in the department, and he chaired the department, what was then the Department of Health and Human Performance, from 1987 until 1990. Prior to his untimely death in 1994, Dean and his wife Sally established an endowment fund in memory of Dean's parents, Harvey and Bomel Pease, who died in 1990 and 91. Dean and Sally chose to direct a portion of his parents' charitable trust to 3 specific programs to benefit students and faculty at Iowa State University. The greatest proportion of the Pease contribution was used to create a Scholar in Residence fund for the department, which is what supports this event. Each year we are able to bring a scholar in our field to campus to interact with faculty and students in the department, both undergraduate and graduate, and of course for this special presentation, the Pease Family Scholar Lecture. I'm very pleased to introduce this year's Pease Family Scholar, Dr. Bradley Hatfield. Dr. Hatfield is professor and chair of the Department of Kinesiology at the University of Maryland in College Park. He completed his undergraduate studies in Canada, his homeland, at the University of New Brunswick, where he received bachelor's degrees in physical education and psychology. He then moved to Penn State University, where he earned his MS and PhD degrees in what is now the Department of Kinesiology. At Penn State, Dr. Hatfield studied under the mentorship of Dr. Daniel Landers, a well-known scholar in sports psychology. And it was at that time that Dr. Landers and his students began to explore the interface between psychology and physiology in sport and exercise applications. And Dr. Hatfield was a driving force behind Dr. Landers' shift towards a psychophysiology focus. Dr. Hatfield's interests are diverse. He's had an interesting set of work and professional experiences blended in with his academic training and faculty activities. Let me give you some examples. He has been an assistant camp director for the Junior Olympic Development Program at the U.S. Olympic Training Center in Colorado Springs. He was a graduate assistant football coach at Ohio University in Athens, where he also earned a Master's of Sports Administration. He was a biology teacher for a short time and coach and director of athletics at the Rothsay Collegiate School for Boys in St. John's, New Brunswick. He was an intern for the Baltimore Colts, and he was a research associate at the Naval Medical Research Institute at the U.S. Naval Hospital in Bethesda. So you can see the experiences that he draws on are quite diverse. Dr. Hatfield began his long affiliation with the Department of Kinesiology at the University of Maryland in 1982. He has risen through the professorial ranks and along the way has added faculty affiliations in the Center of Aging, Neural and Cognitive Sciences Program, and the Center for the Advanced Study of Language at the University of Maryland. As already noted, Dr. Hatfield uses psychophysiological methods to assess the effects of exercise on mental health. His research addresses both the health-related and performance-related aspects of humans in exercise and sport settings. By using both psychological and physiological theory, principles, and research methods, he and his students examine mechanisms underlying the mental health aspects of exercise engagement. He's authored more than 80 refereed research publications, 15 book chapters, 1 textbook. He shared his research through more than 50 invited presentations at a variety of universities, seminars, workshops, and professional meetings. And he and his graduate students and research colleagues have made more than 200 presentations at international, national, and regional professional meetings. His research has been well funded via external grants, both large and small, from a variety of agencies and foundations such as the National Institutes of Health, U.S. Department of Defense, particularly the U.S. Army, American Heart Association, American Federation of Aging Research, and the National Rifle Association. I could continue on with many more accolades, but you came to hear Dr. Hatfield speak. Suffice it to say, Dr. Hatfield is highly accomplished and well recognized internationally as a leading scholar in exercise and sports psychology. And he is also a great speaker. I think you're going to enjoy what he has to say tonight. So it's with great pleasure that I welcome and introduce Dr. Brad Hatfield as the 2011-12 Pease Family Scholar. Please join me in welcoming him.
Thanks, Bill. First question I'll ask is, can everybody hear me okay? All right, great. And now I'm going to present a lot of material, but I'm going to give you a bottom line as we go through a lot of the slides. Details, not terribly important tonight. Big picture. And I'd like to start off with paying tribute to this organ right here. I put on the slide 100 billion neurons, some estimates are higher, 1,000 trillion synaptic connections. This is the seat of who we are. Over here, I put cognition, our thinking, our feeling, our ability to move. But if you think about it, a lot of what I'm going to talk about tonight is memory. And I'm going to put this talk tonight in a public health context about dementia, but I see a lot of young people in the room. And as we get to the final leg of my talk tonight, it's all about young people. A study that we had done recently which shows a great connection between your lifestyle in terms of physical activity now and what happens down the road at a much later stage in life. There is a cost to everything we do, and how we live right now, one of the messages I'll get across, I believe, has great implications for our future. I'm going to talk about the role of exercise, that's what the little icon here is for, in a public health context like dementia, and I'll explain this word in just a minute for those who may not be familiar. But it's really one of building resilience and tolerance to aging and to disease. Essential medicine for some. Some people, because of the genotype that they have, with mother and father and the heredity that they pass to you. Some have, I would say, in this domain of the way the brain works, have more to gain, more to lose by being sedentary. And then, again, because we have such a nice group of young people here tonight, I'm going to talk about something called an investment hypothesis, which relates to the earlier life activity and how it is related to the gracefulness of the aging of the brain. So that public health context, Alzheimer's disease, I'm going to use as a background in terms of the relevance of exercise to the brain. Well, what is it? It's an irreversible, progressive disease that slowly destroys memory and thinking skills. It's probably been around since the beginning of time, formally talked about in 1906 by Alois Alzheimer, a German physician. Notice the map here, the darker colors, there's the North America, Africa, Europe here, Australia. Notice the darker colors are in what we think of as the more advanced, economically rich areas of the world. This is where people are living longer and probably have, because of the way our communities are built, not as active as in many parts of the world. The way this disease progresses is this is a slice of the brain called a coronal slice, just like if a blade came down and cut me in half right now and the front half fell towards you and, well, hopefully the back half will stand up. So your eyes would be out in this area and the back of the head would be towards the back of the screen. And I drew a box around a structure with a little label here, and this is an area called the hippocampus. And this is as if you turned the head to the side, and so you're looking at a temporal view. And in preclinical Alzheimer's disease, we start to see the destruction, and I'll play it out to the full extent. Notice it's nice and healthy here, and we're having a gap or atrophy, and then finally it's gone as our other structures of the brain, like the ventricle, is getting bigger. And notice if you turn the brain to the side, the pathology is starting here and it spreads. And unfortunately over here, the memory of who you are and your loved ones is gone. And imagine not only the great cost to the individual, but imagine the cost to the family. The sons, the daughters, other family members. It does not just affect the individual. It affects many, many people around them. If you look at the statistics here, about 10% of over 65 and 50% of those over 85. Now, I can remember, as many of the young people, I'll never be 85. Yes, all of us in this room, if we're fortunate, we will be there. And this is a very real phenomenon. The only way Alzheimer's disease, this is what are called the plaques. And these are the tangles. I'll explain them briefly. The only time you can be really diagnosed of having Alzheimer's disease is at death on autopsy. So physicians usually talk about Alzheimer's-like dementia from your behavior. But they never can be truly sure whether you really have Alzheimer's disease until the brain is looked at death. This is a neuron, and this is a protein that is positioned such that it's exuding from the membrane. And notice there's like a clipping action with these enzymes. And then they come together outside and they form these plaques. And we're doing it all the time, where the body or the brain is able to get rid of these plaques. Some people do it better than others. I'll talk about that a little bit later. But basically these plaques can interfere with neuronal communication. The tangles are inside the neurons. It's a nerve cell. And see this nice little tube? It's like a transporting highway. There's a tau protein that breaks down, and the tubes break down, and so therefore the communication and the movement of important substances inside the cell is destroyed. So the plaques and tangles are the pathology that result in the death of neurons, particularly starting with the hippocampus, which is the gateway to memory. If I asked you what you had for lunch today, the reason that you can recall that is because of the hippocampus, that structure I showed in the previous slide. But another big point I'm going to talk about tonight is many with significant pathology, could be in middle age, could be elderly, many with significant pathology show no symptoms. They're quite tolerant to it. Now, by the way, before I go on further, right now about 5.5 million cases in the United States. Multiply that by many caregivers as far as the human problem. 16 million projected by 2050. When I was a kid growing up in Canada, the entire nation was 20 million people. 16 million by 2050. 2050, oh, that's a long way away. 2050 is not that far away. Again, think of the number, multiply this by 10 times in terms of those affected, the caregivers. Fifth leading cause of death, the financial burden. Sometimes these numbers are rather meaningless. I'm going to say by 2050, this is probably, notice my plus signs, probably about $1 trillion worth of burden to the United States, and we already know that we have many other financial burdens in the country. If we could delay it, if we could just delay it, and I think that's what the role of exercise is, you could reduce the incidence by 1/3, but you can still see it's going to be quite a battle. And one of the reasons it's going to be a battle is because we're living longer, right? People are living well into their 90s and that. And that's why you see in the more economically privileged areas of the world, it's rising, where people are living longer. If you look at a map, this map is very interesting. The darker the color, the more between 2010 and 2025 we're going to see incidence of it. The lighter color means less of a burden. That's probably because most of these people are moving south, right? People in the particular age groups are moving south. Notice that the burden is high here in Florida and through the Southwest, and getting even higher here in these parts of the world. But in absolute numbers, there would be fewer. This is where the population is heavily concentrated. Now, what's the role of exercise in this? And I have to tell you, if this was 10 years ago, and somebody said, oh, we need to look at how exercise relates to something like Alzheimer's disease, I would have laughed and I would have said, come on. That's like, trying to topple a giant with a pea shooter or whatever. It appears that exercise, what we do in kinesiology, may actually be one of the most promising avenues for dealing with this disease, maybe even more promising than the pharmacological or the drugs. But I'm going to start with this and build a little bit of a story. And then I'm going to go into a part about people who are at different risk. About 20% of us here in the room, I say us, I include myself, about 20% of us in the room here, and I know we all think, well, it's not me, it's somebody else, right? But about 20% of us in the room are at pretty high risk for dementia. And then I'm going to talk about earlier life and how that can be related to this, and then some summary points. Well, to start off with, a very nice big review by the National Institutes of Health. They said, all right, some people are proponents or advocates for exercise, but what's the real meat? What's the real evidence if we walked into a court of law and had to plead our case? Well, there was this thing called the Cognitive and Emotional Health Project. The goal was to identify demographic, biological, psychosocial factors that can help people maintain, enhance their cognitive and emotional health as they grow older. And Hugh Hendry at the University of Indiana headed up this particular study, published in Alzheimer's and Dementia. They identified high-quality studies on the relationship between several variables in cognitive health. Had to have big sample sizes, looking at the age over 65, longitudinal in design, followed over a period of time. One follow-up evaluation, memory had to be looked at, depression, etc. And was physical activity protective if you're not a cheerleader, just looking at the hardcore evidence? And the answer is yes. And what you see here is what really helps the brain to age gracefully, the higher education levels, which we'll talk about something tonight called cognitive reserve. And higher education changes the architecture, the morphology, the structure of the brain. It thickens the tissue density in the white matter tracts, which are like highways between different areas of the brain. These are impacted by education. Socioeconomic status probably tied up with nutrition levels, looking after oneself. But notice I have more physical exercise. It hit the standard of hardcore evidence. And there's so we can make a case, but why? But why does physical exercise do that? Now I've got a study here that I was just shows on a different level of analysis, something called the psychophysiological level. I'm going to show you just some things that we can do. This is an old slide from our laboratory. This is a nice young gal by the way of Dee, was a marathon runner. And she'll look at letters on the screen, and we measure the electrical activity of her brain. And so what she's looking for in a series of stimuli is she'll see an X, an X, an X, an X, and then an O. And we ask her to count the O's. And as she counts the O's, the electrical activity is being recorded up here. And so here's this idea. 90% of the time you see these boring X's, and every now and then you'll see an O, and we look at the brain's response to that. They call it an oddball paradigm because there's an odd, it's an odd balance, I guess, between the targets that you're supposed to pay attention to and the common stimuli. And you go through, by the way, our understanding of how exercise affects the brain is very much dependent on engineers and physicists and mathematicians, because we have to have the technology to get the insights as to what's going on in the brain. I see many times in graduate school in kinesiology, it probably is just as relevant for somebody to have some training in electrical engineering as it is to come purely through kinesiology, although I'm a big fan that they come all the way through kinesiology. What we see here are some responses in the brain every time the O is recorded, and we average them over time. Now, we get a nice signal like this, and this nice big bump relates to how vigorous the brain's response is to seeing the O. So it's almost like a test of neurological integrity. And somebody like Dee, who is a great runner, she can be compared to other people who are sedentary. And what do we see? Well, I'm going to show you some data just from the older high active versus low active people. And here's how it works. This is the front of the brain, this is the back, and so you have a voltage map here. And blue means not much of a response. On this side we have the low fit, on this side we have the high fit. And as we move up more towards the green and yellow and pink, that means a nice vigorous voltage response that the brain gets it when they see that signal. Now notice here with low fit and high fit, about 1/5 of a second after the appearance of the O, there's not much of a response. Not too much of a difference here between low and high fit either. But once we start to get around 300 to 340 milliseconds, we start to see that the high fit people are basically having a more robust response to that particular stimulus. Why might this be the case? Well, we know that there's an increase in cerebral circulation. We know that many neurotransmitters are upregulated with exercise. We know there's the diminishing or the lowering of stress hormones, like cortisol, which have a very negative effect on the structure of the brain. But the big one that's come about, I'd say, in the last 15 years is the so-called neurotrophic effect. And Fred Gage and Carl Kottman's at the University of California, Irvine, have done tremendous work where they've shown that being physically active promotes the release of protein, brain-derived neurotrophic factor, which means brain-nourishing, neurotrophic. This tends to be upregulated during exercise. And this just is in a rat brain, and this is sedentary rats, and this is active rats, and this is also a cross-section. DG stands for a dentate gyrus, which is in the rat's brain structure of the hippocampus, which is the memory area. Now here's my point. Does exercise benefit the animal brain? Yeah. There's a lot of neurobiological evidence that it affects the animal brain. And that neurotrophic effect, this brain-nourishing protein, it increases the synapses, it affects the axons, it puts more fat, myelin, into the axons. You have healthier neurons. That neurotrophic effect in the animal is most pronounced in the areas of the brain devoted to memory. Such an effect in the human brain would hold major implications for the delay. I didn't say the cure, the delay of dementia and Alzheimer's disease. So what about the human brain? Now, the other major player who's done tremendous work of value in the last few years in this area is Art Kramer and his team at University of Illinois. In 2003, they published the first study using magnetic resonance imaging. They took middle-aged people. This fellow here is a little younger than the people in their study. But they looked at people from very low levels of fitness to high levels of fitness, and they looked at the tissue density in the brain. And the aerobic capacity was positively related to tissue density. It appears to slow brain aging. Now, could that be due to these neurotrophic effects like we see in animals? So here what I've drawn is a brain that is it's loose, it's not dense. The tissue density, if you look at this carefully with lower aerobic capacity, is different than the one over here. Here there's greater tissue density in many areas throughout the brain. That was rather a startling finding. But the real need is to conduct a random clinical trial to assess that causal influence, particularly on the hippocampus, and they just did it. They published it this year in 2011. Kirk Erickson, who's now at the University of Pittsburgh, did his postdoctoral work with Arthur Kramer at the University of Illinois. And what you see here is rather startling evidence, I think. The red is a control group that does stretching and toning over a 1-year period, and the blue is a walking group that gets more vigorous over a 12-month period. Here's our friend, the hippocampus, which is the first to be attacked with dementia and Alzheimer's disease. And what Kirk in Art's team showed was a progressive rise in the volume of the hippocampus, both on the left and on the right. And I was listening to Art a few weeks ago at a conference, and this is a real gain. Not just a diminishing of the loss, but a real gain in middle-aged, early older age, in people going through a 1-year walking program. And notice that their colleagues, who were still doing stretching and toning, actually had a normal decline in the volume of the hippocampus. And also what's really interesting is the exercisers had greater circulating levels. They can't look in the brain, right, because of human subject's limitations. But circulating levels of BDNF were higher in the trained group, which strongly suggests that just like in the animal model, we're seeing the same thing in the human being. So this picture I showed before, exercise prescription, it seems what's happening that Kirk Erickson and the team showed is like the perfect antidote to what pathology is doing to this particular region. And mind you, that pathology is not just in people who ultimately are diagnosed with Alzheimer's disease. To a degree, this happens in everybody. In fact, everybody, if we went through a brain autopsy at death, would show plaques and tangles. And yet, the impact of exercise with this neurotrophic effect appears to be right in the area that's most at risk. You know, and I asked Carl Kottman, who did this animal work with the neurotrophins, or is the chief leader of that whole team, why it might be that exercise would impact a memory structure in the brain. And he said, I don't know. And I gave him a conceptual thought, just something very general. I said, could it be that in order for us to survive, that when we move, unlike nowadays where we have cars and highways and signs and supermarkets, and could it be that when we moved, that memory was so key for our survival that you really have to think of it as a system between brain and memory and muscle. You know, we think because of our education of there's the muscles, there's the brain, and never the twain shall meet. But that's not the way the human body works. And so it may very well be that when we navigate and move through space, that there's a natural partnership with the hippocampus and the formation of memory in order for us to survive. So I don't think it's unusual at all that this area benefits from the exercise. Now, by the way, I talk about something called a synchronous benefit. So you have this pathology that's occurring, but at the same time we have this neurotrophic effect, these opposing vectors. So it's not that the exercise is curing, it's that the exercise is giving a benefit in the face of pathology, but it may very well be that the scales are such that the benefit is outweighing the pathology. And that's how you might have the delay of the symptoms. Mild cognitive impairment. This is, some believe that this leads to Alzheimer's disease, but it doesn't always. But anyhow, is a diagnosis given to individuals who have cognitive impairments beyond that expected for their age and their education, but they can still get through daily life. Study was done last year by Baker out on the West Coast, the effects of aerobic exercise on mild cognitive impairment. These were 55 to 85-year-old men and women who suffered, who'd gone to physicians, and their family members are probably saying, you know, there's a problem here. And the fact of the matter is, after 6 months of a trial of reasonably high-intensity aerobic training, they, particularly in the women, not as much in the men, but particularly in the women, they showed improvements in executive function, which are things like concentration, focus, etc., after being randomly assigned to non-exercise or exercise. Now, there's very little that's been done with Alzheimer's disease, and it may be that they're at a stage where really it's too late. But at the same time, this is very promising. And this gets at a model. If you look at this particular figure and you think of cognitive function, how well you remember, how well you think, how well you concentrate on the vertical axis. In green, what I've drawn is somebody who would have high, what I call cognitive reserve. And cognitive reserve is this issue of the thickness of the brain, the density of the tissue, the circulation through the brain. And this hypothetically is somebody who's low, maybe through poor nutrition, less physical activity, less cognitive stimulation. And on my cartoon, I draw the deterioration of plaques and tangles coming in at this point. And notice that this person in the green line, over time, can still function very, very well. And notice that this person begins to drop off. And that's, I like to use sometimes the analogy of a pair of pants. If you had a nice long pair of pants and they shrunk a little bit, they still look pretty good, right? So even though the pants have shrunk, You had so much length to begin with, they still look pretty good. But if you had kind of short pants, you were a little uncomfortable with them, and they shrunk, they look really bad, right? So it's almost like the brain in some people has enough reserve to absorb the pathology. Well, another example I give you is a checking account. The economy's bad, but some people may have built up the reserve in their bank accounts that they're financially independent. So as bad as the economy is, they're fine. There are other people living from paycheck to paycheck. And then when the economy goes bad, they're in trouble. They're the ones who have the low reserve. And much of your lifestyle is building the reserve in terms of the choices that we make. So the term cognitive reserve, or like I like to call it tolerance, describes the mind's resilience to neuropathological damage of the brain. The mind's resilience is evaluated behaviorally, whereas the neuropathological damage is evaluated at the cell level. So, in other words, some people, they look fine, they can tolerate, we could see disease in the brain, and exercise may be building that. Now, epidemiology shows us with studies, here for example, women with greater physical activity at baseline were less likely to experience cognitive decline over 8 years of follow-up, even after controlling for age, education, comorbid conditions. Richards et al. Noted a protective effect of physical activity in middle-aged men and women from earlier participation in their 30s. Again, what we do seems to have an impact later on in life. This notion of a threshold, I put it, implies that aging is associated with degenerative processes that don't result in symptoms until they accumulate to a critical level. So we look fine. We're thinking, we're attending, we're concentrating, we're just fine until we cross that line. And that particular line, I believe the role of exercise is to move that line so that you can tolerate much more before you cross it. And that neurotrophic effect is probably a big factor on that cognitive reserve. Now, the reason I put this, I love this picture. The other hat I wear is studying sport performance or elite performers. It was about muscles and genes, but we're not talking muscles and genes tonight. We're talking about the brain. So I put the consideration of genetic variants to account for variations in brain adaptations. What I want to develop over the next few minutes is the notion that exercise may be essential medicine for some folks. Now, we're talking about Alzheimer's disease tonight, and some are at greater risk for this terrible disease. So we have a gene on chromosome 19 called ApoE, apolipoprotein E. There are 3 variants of it: ApoE2, 3, and 4. And thank your mother and father if you're a 2 or a 3. If you're a 2, you really ought to thank them. Now, you know we get 2 copies of each of the alleles, so you could be like a 2-2, or you could be a 2-3. Or you could be a 3-4, or you could be a 4-4. If you carry a copy, a single copy of a 4, or a double copy of a 4, it puts you at greater risk for Alzheimer's disease. Does it cause it? No, not even close. But what E4 means, if you have 1 copy or 2 copies, it means that the maintenance crew that looks after your brain doesn't quite clean out the plaques as well. And the overall maintenance of the brain is a little bit compromised. That means that if the pants shrink, you don't tolerate quite as well. Or if pathology is there, you cannot absorb it or be as resilient to it. That's why ApoE4 is more prominent in Alzheimer's patients. It doesn't cause it. It just means you're more likely to express the symptoms or cross the threshold if you carry the E4. However, we can control lots of things in our lifestyle to build that reserve, right? Now, this is a study that was reported in Medicine and Science a few years back, a landmark study. I think ranks right up there with Kottman's and Kramer's work. And this shows the risk. The higher this red is risk for cognitive decline over a 3-year period. You have 4 groups. This study was done in the Netherlands. The yellow are people, 2 groups who do not carry the E4, right? They're lucky genetically. The red carry the E4. These 2 plots are relatively low active people, And over here are the high active people. Now notice that the people who do not carry the E4, whether they're active or not, they don't have a lot of decline over this 3-year period. But notice the remarkable difference in the people who carry E4. If you're sedentary, you suffer somewhat of a loss in cognitive function. If you're active, you diminish that loss to a great degree. The lower the plot in this case, the better, because we're talking about decline, so we want to reduce the decline. That's very interesting too. But why? Now here, in terms of the threshold, notice with E4, I've moved the threshold over here to the left. If you carry E4, you're more likely to cross that threshold at an earlier stage in life. But have good hope here, because if you exercise and you make good lifestyle choices, that kind of increased risk can be largely diminished. The role of exercise is rather profound in terms of the E4 relative to the non-E4 because of that earlier threshold notion. Now, neuroimaging is very, very important in terms of understanding what's going on with the brain here, because I put invisible brain alterations may be occurring while behavior is normal. These are the brains of relatively young people here in their 20s and 30s, and what we see here in purple are areas which these are E4 carriers, they carry the E4 allele. They're just sitting there, and what we see is the brain is going through like a lowered metabolism at a very young age. We wouldn't know that without neuroimaging, because they're functioning completely normally. Only through neuroimaging do we start to see at this level these differences in the brain. So a doctoral student of mine a few years ago published a study using neuroimaging in the form of something called magnetoencephalography, and it was to determine if physical activity attenuates these ApoE4 related deficits in cortical activation. And these are people in their 50s to 70s. So here's the kind of task they did. They looked at a cross, and then they saw a series of letters like this. And then, after a brief period, they had to recognize whether they had seen that or not. And then what we would do is we'd look at them in a machine like this, and I always like to joke and say the fraternity participants, not that we had them in this study, this was older, but they always feel very comfortable in here. I don't quite know why, but anyhow. So this machine measures the activity of the brain, and we put older people in here. And those people were in different groups, very active non-carriers of E4, and inactive non-carriers. And this is the groups we were particularly interested in, the active carriers and the inactive carriers. And we looked at how the brain responded when they would do this recognition task. And we had a design where there's your 4 groups: high active, non-carrier carrier, low active carrier, and we followed the brain over a period of time. And what you see is right here, this is the group that carries the E4 and is low active. This is where the stimulus is presented at this point in time, and this is following the brain activity. Here we're looking at it in about the right temporal area of the brain. And we see that the active E4 carrier is similar to the non-carriers in terms of a nice, healthy spike. But the sedentary E4 carriers show a diminished response. Now, mind you, what's interesting about this is they perform the task okay. So I talk a little bit in a few minutes about a forecast type nature. We localized it to what's called the medial temporal region of the brain. And notice here on latency. So looking at the C, this is where the eyes would be, and this is the left and right temporal regions. This group sticking up is the low active E4 carriers. Low active E4 carriers. The higher here means the slower. These are your active carriers. These are your active carriers. This is the left. This is the right. So notice that what you have is the ones who are at risk for the dementia. If they're physically active, they are indistinguishable from the non-carriers who genetically are at lower risk. And we see that on the left and on the right response. So the cortical activation in the physically active carriers during working memory is similar to that of non-carriers who are at lower risk of dementia. Could it be that they are getting, just like Kirk Erickson showed, that they're getting this neurotrophic effect by moving around in space and being physically active, that somehow is giving a protective effect to the memory areas of the brain such that they look just like people who are at lower genetic risk in their age group. And our notion here is that fitness and activity are good for the mental performance, can be measured in reaction time, can be measured in accuracy of different tasks. But the slope of the line is greater in terms of benefit. In other words, at low fitness levels, the E4 carrier doesn't perform as well as the non-carrier, but they're able, through physical activity, to catch up. And this particular slide shows that we can look at genetics, age, education, fitness, and the interaction with genes. And we use what's called a hierarchical regression approach. And we can look at many, many sensors of this MEG, and we can give them different tasks. And we can also look at networking in different areas of the brain as they do different tasks. And we can start to compare those at risk versus those who are not at risk, or looking before and after exercise. By the way, one of the things we found in our studies is that when it comes to an executive challenge with the frontal area of the brain, that as the fitness goes up, so also does the activation of the frontal region of the brain, both in carriers and non-carriers. And some of you in the room may know that the frontal area of the brain, even though I've talked a lot about hippocampus and memory, the frontal area of the brain ages fastest in normal aging. And what we see here is a nice vigorous response when we put them in that MEG device, that there is a nice spike of activation here in the frontal region as the VO2 max goes up. But sometimes, particularly when we go to the temporal area where those memory areas are, what we begin to see is a difference. These are the carriers. This is speed of that electrical response of the brain. And with greater exercise, we see that the carriers improve, and the non-carriers, they're already operating at a pretty high level. They're pretty static. The importance of neuroimaging, too, these developments that have occurred. Now, by the way, once I get through my point here about people who are at risk, then I'm moving into the young age group as I get into my final leg and talk about how the early life activity starts to relate to all this. So just bear with me a little bit. The importance of the neuroimaging is that, again, these invisible things. Susan Buchheimer, who is at Harvard University, now at Johns Hopkins, published, I think, a landmark study a decade ago in the New England Journal of Medicine. She had ApoE4 carriers and non-carriers do mental tasks. And what she showed is if you followed the brain, here's the E4 carrier, if you can see my line on the gray. Notice that the brain response is higher. The brain is working harder in the E4 carrier as they do the mental task, like a basic memory task. The non-carrier shows less of a response, and this is in the hippocampal region. Now, I use the term a forecast model, and here's what I mean. And another woman by the name of Risa Sperling, I think she's at Columbia University. What she's looked at is this brain activation. If you look at risk of Alzheimer's disease, here is an E4 carrier who shows no symptoms. Here is somebody who has this early cognitive impairment. This is late cognitive impairment, and this is Alzheimer's disease. And my line here is normal activation in the brain. Now notice that even though all these two groups right here would look completely normal if you gave them a test of memory, attention, focus. But notice how they're above the normal brain activation line. Now, all of us will go downhill with aging because neurons in the brain are dying at some rate. You can arrest that with a healthier lifestyle. But notice they're compensating, and they're really compensating. And that compensation model means that some people are working really, really hard. even though they're able to get through life and they're able to perform their tasks. By the way, this kind of thing is also believed to go on in things like post-traumatic stress disorder and in traumatic brain injury with military. It may be that they can get through life, the daily events, but the brain is working so hard. And so only through neuroimaging can you see this kind of effort relative to cognitive performance. Now, when the brain gets to the stage where it can no longer compensate, this is where you end up with the physician and you're saying, my God, I couldn't remember how I lost my way going home today. I forgot where I lived. And imagine the horrific state that the individual would be in once they reach that particular stage. And my point to you, sometimes think about the future, is just like we go for blood pressure monitoring now, to forecast the potential of some kind of stroke or some kind of heart disease in the future. I believe with technology that people and their quality of life will actually have neuroimaging while people perform mental tasks just as part of regular medical diagnosis, particularly if we're all going to live to be well up into our 90s. And you could monitor over time how hard the brain is working relative to simple diagnostic tests. And if it is working harder, it may very well be it may very well be a forecast of potential trouble here. This kind of compensation in the face of normal cognitive performance could be indicative of eventually that compensation is going to fall off, and then you're going to have symptoms. So neuroimaging is important. Okay. Now, if you can bear with me, here's my third and final leg before I go into summary. An investment hypothesis, early life activity and a yield on investment, just like we put money into retirement accounts. The effect of physical activity on the developing brain and cognitive reserve, and I'm going to talk a little bit more about ApoE on this too. Now, one thing that you'll find interesting about what we do now and how it's related to the state we're in later. Here's a study a couple of years ago, Journal of Gerontology. Exercise at midlife may reduce the odds of dementia in older adulthood. This is a verbatim quote from the paper, suggesting that exercise intervention should be explored as a potential strategy for delaying disease onset. That's delaying that threshold and contributing to cognitive reserve. We found that participants who engaged in light or regular exercise had reduced odds of dementia and AD approximately, that's not a typo, 31 years later. 31 years later. And I think there's a great parallel to the religious order studies. This was a group of nuns and priests who, many of them lived to be quite elderly. And because the significance of the studies is many of them have a very homogeneous lifestyle. So things like income and things like stress are largely evenly spread because of the relative sameness of the lifestyle. And this is David Snowden, who's done work with Riley and looked at the relationship of early life mental abilities and activity and what happens in really old age. Check this out. Early life linguistic ability and late life cognitive health. And I think it's a nice model for exercise. Early life linguistic ability. Around the age of 22 years, quite young, probably very similar to the age of many in this room, there were autobiographies written by the women just getting cloistered and coming into the nuns. And they looked at the idea density. In other words, why do you want to be a nun? Well, some people must say, I don't know, I got nothing else to do. Period, right? And some might go on with a very rich narrative in which they explain in detail and with enthusiasm. It comes out in their language and in their writing. Early life idea density was positively related to brain weight, inversely related to cerebral decline or atrophy at death. Low linguistic ability in early life may reflect suboptimal neurological and cognitive development, less reserve. And check this slide out. This is low idea density, and this is the percent with Alzheimer's disease from the autopsied brains. And you can see about 65, 70% high idea density. So what's happening here is the high idea density is probably a proxy. Remember I put up 1,000 trillion synapses on my very first slide? And the more that you have those synapses and that rich communication between neurons, which neurotrophic factors directly have a significant and remarkable impact on. But that high idea density would give you the reserve, hypothetically, or it's a proxy for reserve, and therefore the brain becomes more resilient to the slings and arrows of decline with age and pathology. To me, that's a rather remarkable illustration. Now I want to put it in my last study I'll highlight here about with exercise. And this was a study done with a student of mine just in the last couple of years, and John Polich, who's at Scripps Research Center, Stephen Roth, who's in our department in the genetics area, and myself. And we use the title, Cardiovascular Fitness Modifies the Relationship Between Genotype and Neurocognitive Function in Late Adolescence. In other words, when we take, in this case, it was college men, which I'll profile here in a minute, but the same would apply to the college women. In fact, the women in the room should take solace in the fact that women seem to have more of a beneficial effect in the neurocognitive domain than men do. And it may have to do with hormonal differences. I'm not quite sure. Now, the reason that we were interested in the effects of exercise in the young person's brain is, get a load of this. There's continued frontal lobe development after puberty. Prefrontal, so right where my forehead is, prefrontal cortex continues to myelinate, get the nice white matter and establishing those tracks. Through the third decades of life, or around 25 years of age, synapses in the prefrontal lobe are pruned, they're initially overproduced and pruned up to the age of 30. That means that the frontal area of our brain, another critical area to aging besides hippocampus, doesn't even fully develop till we're around 30 years of age. Now, I always like to say to people in that way too, I've gone to a few leadership conferences at West Point in the last few years. And they piped in the last one I went to, they piped in some of the young officers from Afghanistan. And these are people dealing every day with life and death issues. No joke, right? They're 22 years of age, 23 years of age. And they're asked to make critical decisions that can impact the actual life of people around them. And notice that we know from neuroscience that the human brain isn't even fully mature in terms of this frontal area, this executive decision making, until about age 30. But it's a very, very plastic area of the brain. And could physical fitness actually be beneficial in such a plastic period of development? Prefrontal cortex is the brain area that develops most, but also last in the course of development. And it may very well explain some of the differences in adolescent behavior rather than just culture. It may be that some of the differences between adolescent behavior and later in life is actually due to the incomplete neural development of that area. So your lifestyle, sleep, exercise, nutrition, etc., may be laying a critical foundation in this very plastic developmental period. Now also notice our good friend ApoE, not just relevant to elderly, but relevant to people in every single age group sitting in this room. Notice between the ages of 20 and 39, 19 and 28, and children and adolescents. Glucose metabolism, here's your E4 carriers. Notice the lines are lower than NC, which is non-carrier. Glucose metabolism, again, here as your E4 carriers over here compared to your non-carriers. And here we have the E4 carriers in terms of cortical thickness. This would be the E2 carriers, this is the E3 homozygote, and this is the E4s. So the brain looks like it doesn't quite have the reserve, even at a young age in life. Carrying the E4 allele negatively influences. So what did we do? Then we went to this particular age group and we looked at E4 carriers and non-carriers, and we looked at fit ones and we looked at unfit ones, and we looked to see is there actually a difference in the function of the brain when we challenge them with simple mental tasks like what I talked about before with the oddball. So, in other words, does it make a difference, even at that age in life? So the purpose was to examine if cardiovascular fitness modifies the relationship between this risk and neurocognitive response during late adolescence, or you could call it early adulthood, right? And what's really interesting is, I think, is if we start to take this down and look even younger. And we know that children vary very, very much in terms of how active they are. So we had high-fit carriers, low-fit carriers, high-fit non-carriers, and low-fit non-carriers. They're all around 20 years of age. You can see the weight, pretty normal. These were males. These are in pounds, 181, 171. Estimated VO2 max are fit. You have 52. Not super athletes, but pretty good, versus 36. And in our non-carriers, the fit were 54 mLs per kg and around 40. And this is an intelligence test, and obviously they were all normal. And this is the technology we used. And basically we're looking for the amplitude of the brain when we show it a basic challenge. And we're also looking at the latency of these waveforms. Now, for instance, if this is where you see, if you're presented on the computer screen these X's and O's and you're told to pay attention to the O's, or sometimes you're shown X's and O's and you're told respond to the X, respond to the X, respond to the X, withhold on the O. That's called a no-go task. And that no-go task is particularly sensitive to the function of the frontal region of the brain. In fact, my friends, when we imbibe on alcohol, one of the reasons our behavior changes so much, many people in the room may already know, is because there's a direct effect on the frontal lobe of the brain. The frontal lobe of the brain inhibits our behavior in social situations. Notice how we become less inhibited. I've only heard from my friends, not from direct experience, right? But notice that we become more gregarious. We become less inhibited when we take alcohol. That's because alcohol has a very direct effect on the frontal region of the brain. Now, but inhibitory behavior is not just social inhibition. Your ability to concentrate and focus, your ability to listen to me and focus out all kinds of other things, it has to do with the frontal lobe of the brain. When we see a nice peak like this, it shows a nice response. If it's early, like if this is where the X or the O was shown that you respond to, this is the time between the presentation and the hump right here, which is this attentional allocation. As you get older, the latency gets longer and the amplitude gets lower, and so we can use it as a nice neurological measure of the integrity of your brain. And what we have here is, here's our carriers, here's our non-carriers, here's the high-fit people, the low-fit people. We're going to look at different brain regions. We're going to look in the frontal, the central, and parietal. The frontal, the central, and the parietal, which is about near the crown of my head. We're going to look at those different brain regions in low-fit and high-fit, and within low-fit and within high-fit, carriers and non-carriers at that nice, young, healthy age of life, 18 to 22. And here's what we found. Now, I like to do this. I like to do expected findings, and then I like to show you what we found. So I go expected, observed. Now, on the amplitude, here's what we expected to find. We expected to find on this no-go task where they would press at the X, press at the X, press at the X, press at the X, and then when that infrequent O come up, don't press, withhold the response. Because that involves the frontal region of the brain, these are the carriers, these three lines. This represents the frontal area of the brain, the central, the parietal. These are the non-carriers to the right. So here's your risk group, here's your non-risk group. In the darker color is the high fit, and in the gray is the lower fit. And what we expected is that high fit people, during this kind of a challenge, high fit people in the carrier group would be distinguished from their sedentary colleagues and show a nice vigorous response in the frontal region of the brain. Could we pick this up at this tender, young, healthy age? And also we gave them the task that the Lady D, I was showing you much earlier, did, in which there was just press the button when you see that infrequent O. It's not as much of a demand on the frontal region. We figured there'd be no difference because these people are all young, they're all healthy. But here's what we found. What we found whoops, let me go back that one. What we found on this amplitude right here was a statistical interaction between the genotype and the physical fitness and the region. And so regardless of the task, notice the task is not in here, genotype, physical fitness, region. Regardless of the task, what we found is in the frontal region, the high fit young men showed a superior response relative to low fit in both the frontal and in the central areas. Notice in the non-carriers, high fit, low fit, they're indistinguishable. And that probably means that they have pretty good integrity of the brain, and no matter whether they're fit or low fit, there's kind of a ceiling effect there. Everything's young, everything's healthy. But in the carriers, of which about 20% of us in the room are, and here we actually match the numbers, you have a more vigorous response in the carriers who are fit. Now, the good news is, my friends, this is what we thought would happen with the latency, which gets at speed, how fast you are relative to your fitness and genotype in this adolescent period. We thought that the higher here means slower. We thought that the low-fit carriers would be slow. We didn't see differences in other areas of the brain. And with our non-carriers, we'd see no difference. And over here on this task, since it doesn't relate so much to the frontal lobe, this critically developing area, there'd be no difference. What did we find? The good news is we found that in both tasks, regardless of genotype, everybody benefits, regardless of genotype. And here's a summary statement for you. On amplitude, E4 carriers may derive particular benefit from fitness. Okay? Something's happening that you can't really pick up if you just give them a memory test or you talk to them. You can't really pick it up, but you can pick it up with something like the event-related potential. Cardiovascular fitness in E4 carriers may be protective against the susceptibility to the liabilities, hypometabolism and cortical thinning. And those other studies that I showed you, people like Eric Ryman, superb scientist, who showed that young people had this hypometabolism. And the work on the cortical thickness, I forget the primary investigator right now, but done at NIH. We know that this happens with young people. The fitness seemed to have an effect in our study. In terms of the latency, the good news, everybody benefited. Everybody benefited. But latency and amplitude are getting at somewhat related but different processes. Okay. Now, you can probably say, oh, good. He's got to his summary slide. Okay. What have we learned in 14 years since the Surgeon General came out with a report in 1996? Now, young folks in the room may think 1996 is a long time ago. It is not. In the landscape of science, it's like that. In geological time, it doesn't even exist. But it was 1996 that the Surgeon General put out a report on physical activity and health. Now, there have been some more recent ones from Institute of Medicine, which comes under the National Science Foundation and that. But this was a landmark report. If we were back in 1996 and could project ourselves 15 years into the future, and here we are, what have we learned? An awful lot. The linkages between physical activity and cognition are robust. There are viable biological mechanisms to explain this. There is biological plausibility, which is important for proof. There is some degree of task specificity. In other words, things that deal with memory. Not so much the long-term memory, but the formation of new memories with the hippocampus. And also the frontal executive with focus and concentration. My mother, God rest her soul, I remember when she was in her 80s, used to tell me, I used to say, Mom, why don't you read a book to feel really good and relaxed? And she'd say, Brad, I just can't concentrate. I can't read the book like I used to when I was younger. My mind just can't focus on it. And that's largely a result of changes in the frontal region of the brain. And thanks to Dr. Kramer, who I couldn't highlight too much tonight, we also know that there's a big impact on the frontal region. So, executive tasks, memory tasks seem to particularly benefit. By the way, long-term memory doesn't seem to have a whole lot of relationship. Who was your first grade teacher? Right? What was the house that you grew up in when you were 3, 4 years old? This doesn't seem to be affected too much. It's, what did you have for dinner last night? Where were you last week at this time? What is tonight? Thursday night? Where were you last Thursday night? That is what the hippocampus gets into. And that neurotrophic effect seems to affect that. We need to consider individual differences. We don't all come from the same cookie cutter. We need to consider the gene-environment interaction. Multi-level measurement is critical. We can look at a person and we could put them through exercise compared to another group that's sedentary and not even see a difference when you just challenge them at the cognitive level. Although many times you still will, but we might not see it. But what we might see is that the one with neuroimaging who's gone through the exercise actually strategically performs the task in a much more efficient manner. I will tell you right now that, the compensation thing that I talked about, the fit one may be using minimal resources. The one who's of lower fitness may look fine. You say, well, Joe or Mary, they're doing just great. They can think clearly, they can remember clearly. But what we find is a very expensive process in the brain. It's working very hard. That's why we need to use multiple levels of measurement. The future, 2 slides on the future, then I promise I'm done. Lessons from the NUN studies. Study physical activity and brain development in children. Because what's happening with the children may be laying the critical scaffolding for the future. It may be a critical period that we can never recapture, just like bone mineral density. If there is some kind of deprivation of weight bearing and running, there is loss of bone mineral density that you cannot gain back later in life. It is within the realm of possibility the same thing's happening in the brain. We need to conduct studies to examine the influence of exercise on brain regions like the hippocampus, which Kirk Erickson did, but in genotypes like E4. And I'm sure that that's probably being done in somebody's lab right as we speak. And that's a good thing. We need to study clinical populations. AAMI looks like an automobile company, right? But it stands for age-associated memory impairment. MCIs and ADs I talked about tonight. We need more studies on clinical populations to see if we can slow, not cure, but slow the progression. Maybe slow it to the point that it never reveals or never presents. And that would be quite something. That's the same as a cure. Investigate neurocognitive benefits based on community design. I think that, when we have communities that people move in, that physical activity may save our country not only grief from loved ones and caregivers and those who become afflicted with aging decline, but it also may have big impact economically for the country. There is a need to assess the dose-response relationship between physical activity and impact on areas. Some people ask the question, how much do I need to exercise? My answer to that is any kind of exercise is good. How good? We don't really know. But anything you do, even swinging a golf club, they've done neuroimaging studies that show swinging a golf club affects the architecture of the brain. But is that necessarily the impact that has on the brain? Is that going to pay off in terms of some kind of affliction that we might have, like memory loss? Probably not. But anything that we do, just walking, anything that we do is going to have an impact on the brain. We always thought of this as an inert organ that just died over time. Now we know that it's very dynamic and it can change within seconds. It can change within minutes. So I think that the dose response is something that's going to be very hard to get a handle on, but it is something that will be addressed in the future. We need to examine the moderating effects of stress. I can't tell you, those neurotrophins, that thing I talked about, BDNF, brain-derived neurotrophic factor, it's very much interrelated to other processes in the brain, like cortisol. And so therefore, we don't exist in a vacuum. The stress could very well undo many of the benefits we get from the physically active lifestyle. And this big one here I've done again. We need to examine, I really think this is a major thing in kinesiology for the future, to look at the children. Built communities, children, and this work by Hillman at University of Illinois is some of the first psychophysiological studies with children, and they're finding with fit children versus lesser fit children, and some of the studies by Davis out of the University of Georgia are very compelling that there is an impact on children. There is an impact on adolescents. There's a greater impact on those who have certain genotypes. and it can build this particular reserve. If you look at obesity in the country, it's prominent. So I got this slide from Art Kramer, our challenges before us. There's much work to be done. And with that, I would simply like to acknowledge a lot of colleagues and end on a note that I find is very important for our field. We have a very broad field, from philosophy to neuroscience to cellular processes. That's the beauty of kinesiology. It covers everything. I say, when I talk about our Department of Maryland, it covers from the cell to society. The ancient Greeks had a notion, mens sana in corpore sano, which roughly translates to a healthy mind in a healthy body. And I put at the bottom from philosophy to science, because the science is really bearing out that this is good medicine for the mind. The ancient Greeks were wise in this philosophy. Different students of mine at the University of Maryland, colleagues. One of the great advantages we have at the University of Maryland is being in the Baltimore-Washington corridor. Johns Hopkins, Georgetown University, George Mason University. All of these people have had some hand in some of the things that I have presented up here tonight. And it looks like the computer wants me to stop. So even though I know you want me to go on for another hour, I will stop right here. And I would like to say that I really appreciate Dean Peace and the gift that he made to the department to allow me to come out and share some things here with you tonight. And I'd also like to say, as a department chair, and I understand you said that he was department chair and you're a department chair, it sounds like he's made quite an impact on the Department of Kinesiology here with both. It's a very difficult position to lead a department, and for him to make a gift like that means that he gave a double tribute to the unit. And I would like to thank the Peace Family Fund very much for coming out here tonight, and for you taking your time and listening to what I have to say. So thank you very much. And that says we've crossed the finish line.
Are there any questions for Dr. Hatfield? We'll take a few minutes for questions.
Yes.
Going back to the study that was looking at liability and cholesterol, do you know of anything that relates to the use of somatostatins and possibly reducing your cognitive tolerance?
Theoretically, the question was about somatostatins and whether that would have some impact on cognitive function. I don't know specifically. A couple points, though. Theoretically, it would seem to make some kind of sense that even though I talked a lot tonight about neurotrophic effects, there wasn't enough time to really talk about angiogenic or vascular or circulatory responses. So it would make sense. I will tell you also that another big affliction besides Alzheimer's disease is stroke. And what is the impact of exercise on stroke and circulation? And there are studies just beginning to come out of the University of Maryland at Baltimore on this in this area. So I would say at this time that it would be a guess, but it's not that long ago that it would have been a guess to say that the research was relevant to Alzheimer's disease and this decline like of hippocampus. So no, I don't know of anything specifically, but I would say that that's a promising area. And by the way, the studies that have just come out on exercise and stroke and recovery from stroke, Fred Ivey at the University of Maryland at Baltimore, under the direction of Richard Macco, those studies are showing now that there is a benefit to recovery of function and increased activation in the brain of stroke patients. But I'm sorry I can't give you more detail on your question. Uh-oh, a former Marylander.
Thank you, Dr. Hatfield. From your presentation, seems to me most evidence supports that exercise, especially cardiovascular related exercise, is beneficial for the non-carriers, the APOE, E4 carriers. A simple question is, how do I get my genotype tested?
It's not hard. There are ethics involved in terms of the entities or organizations that will give it to you. It's a very simple test. It can involve, doesn't even have to involve a blood sample. It can be a cheek swab to get that. The issue becomes, is one well served to know that? Because this stress that can accrue from the worry of knowing that you have an increased genetic risk could be a problem in and of itself. Now we get into the realm of philosophy or even ethics, and my sense would be that one is best to adopt the personal philosophy of thinking that they are vulnerable and to adopt a lifestyle where they would practice healthy behaviors in a preventive type role. Because here's the other point. You might say, yeah, but if I know that, then I'll be extra motivated to get my exercise. And I have a colleague who's a double E4 who knows it. And I wonder sometimes if he doesn't worry more about it, although I will tell you he practices a very physically active lifestyle. But that's a tough one. I would say that it's not hard to find out at all whether you're an E4 carrier. But I would say it's much more difficult to make the decision whether you would want to know or not.
Very interesting talk. Sounds like the interaction between the alleles that a particular individual carries and the lifestyle that they have has a huge impact on whether or not you develop the disease or how soon you develop it. Is that correct?
I would still say that what I have presented points strongly to that, but as anybody in the room and the students and researchers in the room always know that there can be challenges to absolute certainty in these kinds of things. But I would say it's a reasonable picture right now that there are individual differences and some would have more of an apparent benefit to gain than others.
Sure. Do you have any idea of whether, and I know estimates have to be taken with a grain of salt, but whether there are legitimate estimates for heritabilities, what the extent of genes play in developing Alzheimer's, what the extent that nutrition plays, and then what the extent that physical activity plays? Has that been measured statistically? Well, for instance, you have epidemiological studies which have looked at genetic status, and they have looked at physical activity patterns, although epidemiological studies, it can be very crude, the way that they're measuring physical activity. And then they do very quantifiable risk ratios. And there are many studies, and some of them I had just the bylines up there when I had the epidemiology slide. But you have a number of them converging that indicate not only that is E4 puts you at significantly greater risk, but that there is evidence that exercise reduces this. There is one study that appeared in the literature which I think caused a lot of confusion. It was by an epidemiologist at Johns Hopkins University, and since science is in the public domain, it was a study by a researcher by the name of Podewils et al. And Podewils found that E4 carriers did not stave off a diagnosis of Alzheimer's disease if they were physically active, but non-carriers did. And I thought, but Podewils missed the point. We're not talking about whether exercise would prevent Alzheimer's disease. I think I said on several times up here, it does not prevent it, but it can delay. So the particular approach that they took, I think, caused a lot of confusion in the dialogue here. The role of exercise is simply to build the resilience of the brain to make you more tolerant. If you lived long enough, and you have the pathology in you, and you're an E4 carrier, I believe that if you lived long enough, the E4 carrier is much more likely to show and be diagnosed with Alzheimer's disease. However, and this is great you've asked this question so I can reinforce my point. If they did not exercise, the diagnosis probably would have come much earlier. But when you ask me if the things I'm saying, that it's known for certain and is there definitive proof, you always, as an investigator or as a researcher, always have to say, we provide support for models, we provide support for concepts, but typically in science you never have dead proof.
Sure.
Okay?
Yeah.
Thanks.
Thank you.
Maybe one more? Is there a relationship between depression and the development of Alzheimer's?
That's a good question. Now, many of the studies screen for depression so that they can isolate on the impact of the physical activity or the exercise. The depression would imply, in many cases, there's a reduction of dopamine in the brain. I don't know if that's universal, but it's reasonable to speculate that many people would have reductions in dopamine. The various neurotransmitters in the brain, the best answer I can give you is things like acetylcholine and the catecholamines that are in the brain, epinephrine, norepinephrine. They're all intertwined. And I know that some specifically have been shown to be intertwined with BDNF. So it's reasonable to speculate that depression could be associated with a reduced neurotrophic effect in the brain. And many times with depression, as my hippocampus tries to think up here, there is an elevation of cortisol. The HPA axis is heightened. And so I would say that the depression could mitigate against some of the effects, but we also know there's tremendous evidence that exercise has an antidepressive effect. I don't think exercise is a panacea. It can destroy your knee joints, right? We can have orthopedic insult from it. There's a dose response with exercise. I believe we can get too much, but it's probably one of the best darn forms of medicine that we have. So it would affect both the depression and it would affect the memory and the neurotrophic effects. Thanks for asking that, Phil.
Very good. So let's thank Dr. Hatfield one more time.
Thanks a lot.
There's some refreshments back at the back table. Everyone is welcome. First come, first serve.